did her undergrad at UC Santa Cruz, and interestingly studied shark ecotoxicology. And then her PhD with Dave Ethel at Stanford. And she worked on early sea urchin development there. And then she went on to San Diego State, where she had a NSF minority postdoc fellowship. And she was working with Forrest Rauer, and that's where she started moving. And then she held an assistant professorship at Florida International University, and then finally moved to where she is now, which is Oregon State University, where she's an associate professor. And her work covers virology, microbiology, metagenomics, and marine disease ecology. She's funded by a number of different organizations, NOAA, NSF, Oregon Sea Grant, and then the work that she is funded by NSF Dimensions of Biodiversity and NSF Oceanography. So.
Thanks, Katie. Well, thanks for inviting me. It's great to be here at UCLA. I've never been to this campus before. It's really beautiful, although being from Oregon, I'm a little disappointed in your weather. I've been to Southern California 4 times in the last 2 years, and it's rained every single time. So I guess it's me, maybe not you. So if you ever get some rain, maybe I can come down and bring you some. Bring it down from Portland. So thanks for having me. So today I'm going to talk about 2 main topics. I'm going to talk about coral microbiology, who's there, how we've assessed in a phylogenetic framework what kind of microbes are associated with corals, and how the environment might play a role. In the second part, I'm going to talk about an empirical experiment where we changed environmental conditions underwater for a 3-year time series to evaluate how both top-down and bottom-up factors drive changes at the macrobiological scale and also at the microbiological scale. So this is drone footage of one of our field sites in Moorea. This is meant to exemplify a number of the factors that are important when studying coral reef biology. This one is the very optically clear sea surface. You can see straight through this water. French Polynesia has some of the prettiest water in the world. Very, very optically clear. And it's optically clear because there's very little nutrients in the environment. The other things are these dark shapes under the water. Those aren't rocks, those are corals, and they provide the foundational structure of most tropical reefs. In fact, although coral reefs comprise less than 0.2% of the entire Earth's surface, they are thought to contain over 1 million species, or about 25% of the known and described species in all the oceans. So that's a lot in one very small type of habitat. So we're very concerned about the losses of these habitats, particularly their foundational organisms, the corals that provide the structure and food for the reef, for biodiversity reasons. But we're also very concerned about their losses for economic and social reasons, because they can provide up to $30 billion annually to our world economy directly, but also about $300 billion indirectly through things like ecosystem services and cultural services. My lab, there are many threats to reefs. As can be shown here, there's water quality issues like turbidity, things like crown of thorns starfish, which are predators of corals. And one of the things that I study, pollution or fertilizer runoff. There's also physical damage issues, such as those involved in overfishing, tourism and storms, and of course issues associated with climate change like warming seas and ocean acidification. My lab studies 3 of these main factors: overfishing, thermal stress due to ocean warming, and nutrient pollution. And we like to say that we mainly study fertilizer pollution and overfishing, but you can't do experiments these days in the field without remembering that in the background we have climate change occurring. So there's always some aspect of thermal stress that is going on during our field experiments. So while we can manipulate these 2 factors very easily, we cannot manipulate this factor in the field, but we can in the lab, and then we can track this as it occurs in our experiments naturally. So I'm a microbiologist. I'm actually really a molecular biologist, so my main emphasis is using omics to evaluate the bacteria and viruses associated with many different organisms. I've particularly focused on corals. My family's from the Caribbean. I know I look it. My family's Dominican, clearly Dominican, but we're from the Dominican Republic, and I grew up going to reefs. So it's a real passion of mine to try to preserve the reefs, both in the Caribbean and worldwide. So we use omics. We mainly use the presence of nucleic acids and use them as a way to track how bacteria and viruses change in their diversity and in their metabolism in our different host systems. So we use two main approaches. We use gene marker approaches like 16S amplicon data series, where we look at a single marker of DNA, that is the 16S for bacteria, or the 18S for other eukaryotes, or the ITS2 for some Symbiodinium, the algae that are associated with corals. And then we look at the changes in these target pieces of DNA and characterize those communities. My expertise is actually shotgun metagenomics, where we take whole communities of bacteria and viruses as represented by these circles or straight lines. These are their genomes. We shred all those pieces and all those genomes into pieces, and then we categorize those pieces using computer programs to do it. So I have this little dolphin here because although we get to go to beautiful places, most of the time we just sit in front of our computers using algorithms to analyze these data. So I'm going to talk to you about corals today, but I want to point out that I also do other animals, as shown here by Ace Ventura. We also work on mammals now. We have a large Sea Grant funded project to work on seal stranding. So there have been a number of stranding events of baby harbor seals, and we've been using omics like metatranscriptomics, and now we're doing basically straight up genomics to look at the associations with different viruses and bacteria with different stranding events of these marine mammals. But I just wanted to show that because I know there's some marine mammal people in the room. So, so we're going to talk today about the microbiomes and viriomes of corals. And when most people think about when they think about symbiosis of corals, they mainly think about the conspicuous symbiont with corals, and that is Symbiodinium. So this is a coral polyp blown up here, and you can see these dark spots within the coral tissues, which are normally clear. These dark spots, these are Symbiodinium, photosynthetic algae. And this is, they can contain a wide variety of these Symbiodiniums, which if you look taxonomically about the order level differences. So they can contain a wide variety of these Symbiodinium. And importantly, it's this relationship between the Symbiodinium and the coral that allows corals to live in these nutrient-poor habitats, because as they provide metabolic nitrogen to those algae, which can't grow without nitrogen, through photosynthesis, the algae then provides sugars. And it's this relationship that provides the real engine of coral reefs. But back in the late 1990s, people started thinking about other kinds of microbes that might be associated with coral reefs. So if you look at a coral reef here, we know very well that there are these dinoflagellate Symbiodinium that are associated with different kinds of corals. But back in the 1990s and early 2000, it was discovered that there's also a diverse association of bacteria. Along with bacteria, there are other eukaryotes like fungi, apicomplexans, and a variety of other protists. And there are also some archaea, although less is understood about the archaea than probably any of the other groups. So this is what we call collectively the microbiome. And so when I'm referring to the microbiome, I'm usually referring to this whole collection, but in most of today's talks, I'm only going to talk about the bacterial fraction of the microbiome, because that's my expertise. At the same time, there's another whole set of biological entities that are very important for this system, and this is what we call the viriome. And it's composed of both phage, the viruses that infect archaea and bacteria, and eukaryotic viruses that infect the other members of the microbiome, as well as the corals themselves. And a lot of my early work, most of my postdoctoral work, was focusing on describing these different kinds of viruses that are found in corals and to determining their role in coral disease. So I'm not going to have a chance to talk about that today. I did have a stack of 20 slides at the end of this that if you want to talk about, we can talk about later. But they are an important component, especially in terms of regulating the microbiome because they infect the microbiome, but also in regulating coral disease and bleaching. So the main question that we really ask in our lab is, what is the role of this microbiome in coral, animal, and reef health? And how is the microbiome shifted under different environmental and anthropogenic stressors? So importantly, it was noted that different corals contain different microbiomes. So this is a very simplistic slide. If you look at the coral of Parietes, you'll notice that the composition of these different phyla of bacteria are distinct from that from, for example, Apostolopora, a very evolutionarily distant group of corals. At the same time, if you look at fungi, it also has a very distinct microbiota. And this was observed back in the late 1990s, and as a result of the differences in these composition of microbes, it was suggested that the microbiome, because it's species-specific, must play important roles in the health of corals and their resilience, perhaps, to environmental factors. And so a lot of work has been done on that for the last few years, and I'm going to talk to you about an experiment to kind of confirm that hypothesis. So what do we know about microbiomes and reefs in general? We just did a recent meta-analysis looking at the microbiological diversity associated with reef organisms. So if you look at corals here, these bubbles are to represent the diversity of the microbiome. You can see that corals contain a diverse community between hundreds and thousands of different types of bacteria within the corals. However, you can see that in fish guts, it's about an order of magnitude higher. And in macroalgae, even higher. And then you can see in the sediments, it's a fairly high diversity as well, and then sponges, low diversity, etc. So often people will say, if you go to meetings, that corals have some of the most diverse microbiota communities ever, and it's written in like every paper. And it was kind of a bee in my bonnet. It drove me crazy when people would say that, because I'm like, there's no evidence for that. Why do people keep saying that? So we did this meta-analysis to prove that basically that's not true. And it's actually pretty interesting, because if you think about the function of these microbes, if you have wildly diverse microbiota in your gut or on your skin, that kind of suggests that they're not really there for important reasons. So we actually think this is interesting evidence to support the fact that these microbes have been winnowed down to be a selective community because they provide important function. Now, what is that function? Well, for those of you who are not microbiologists, if you go back in ecological history, if you look at what people used to think about microbes, this is a figure that we augmented from Lindemann in the 1940s, whose original hypothesis about food webs contained bacteria in this kind of black box. And this black box contained bacteria, and it was thought that, well, bacteria must be the source of most food webs, at the base of most food webs, in addition to solar radiation. But other than that, it wasn't really clear what bacteria did, although they knew that had some sort of role. Now, we have this image that bacteria do all of these different things for organisms. They can actually drive multicellularity. They can be nutritional symbionts. They can be a direct food source. They can protect animals from predators. And they can also modulate things like the immune systems of animals. Now, for corals, one of the big hypotheses has been that bacteria provide some sort of resistance to environmental change, or thus a resilience of those communities to stressors in their environment. And that's a hypothesis that had never really been tested, because most people love their pet projects, right? I work on a cropper, or I work on sea lions, or I work on rhizobium in one kind of plant, right? So I got to kind of be in my bond. I thought, if we really want to address this question, we have to do it in a phylogenetic context. So we started this project called the Global Coral Microbiome Project. That's real aim was to evaluate the microbiomes across the diversity of corals. And this would answer some question for us. What microbes are found in this whole diversity of corals? Have they co-evolved with their hosts? And thus, what can we infer about their contributions to the health of their corals? And that's what I'm going to talk about first. And then I'm going to talk about this second project, which is really trying to test these hypotheses. How are microbes changed in the environment if we force particular stressors on them? And how does that lead to coral decline? What is the mechanism behind this? And so we love acronyms in my lab, and so this is the GCMP project, or the Global Coral Microbiome Project, and this is Project Herbivore, which I can't remember the whole acronym because it's really long, but it's about removing fishes and adding nutrients to the environment and seeing how corals change. So let's first talk about the phylogenetic inference of what microbes are associated with different groups of corals. This project began in 2014 with my co-PI Monica Medina at Penn State. She is an evolutionary ecologist. She studies the evolution of corals and the evolution of microbes. And led by this main postdoc, Jesse Zanneveld. This is my postdoc here. He comes from Rob Knight's lab. Really fantastic computational scientist, and an evolutionary biologist. And then our other postdoc, Joe Pollock here, who also studied coral disease for his PhD in Australia. And then my very, very talented evolutionary biologist, Ryan McMines, who's graduating soon. So, this is a slide we always like to start with to remind people how old corals are if you're not a coral biologist. So, some perspective. I say here corals are old-ish, because people often would say, well, that's not old compared to my system. But they did evolve. Scleractinian corals or stony corals evolved about 370 million years ago. They split up into two main phylogenetic groups, one group called the robust clade and one called the complex clade. And this is equivalent. I like to put this graph here because it shows about, to give you some perspective, this is about the length of time that it took for land plants to evolve. So land plants appeared about 370 million years ago. And just to show you how they evolved, flowering plants began around here after the split up of the robust and complex clade, and then nitrogen-fixing plants later, after most of these corals had already evolved. So it's a long history. And so one of the things when people say that microbes are important for corals, they're putting all of this very diverse groups of corals that are very old in the same box. So I want to say, well, is that true? Can we actually make blanket statements that say microbes are associated with all of these different corals? So to do that, we had to go on a whirlwind tour. There are 21 coral clades here, and these are the places we used the Census of Marine Life to do this meta-analysis to figure out where all of these different clades were found. So these are the sampling locations all around the world that we had originally targeted, and then reality set in, and these are the places that we went to. So this is a collection. You can go to our website, you can click on every one of our data. All of our data is freely available immediately when it comes online. So these are the places that we sampled and the number of different samples that we've collected. You can click on any one of these bubbles on our website and it'll take you to that, it'll take you to the zoomed in image. It'll take you to that reef. Then you can look at the individual coral samples here, and then eventually you'll be able to click on all of the metadata and all of the 16S microbial community data once it's finished. The data that I'm going to talk to you is exclusively about Australia because it's the first data set that we collected, because it's approximately 3,700 DNA samples, and we wanted to start small. So before we tackled the 3,700 samples, we wanted to see if we could get some information from just about 700. So corals, although they're very simplistic, two-cell layer animals, they do have different, what we call microbial compartments. So this is a drawing of a coral polyp here. There's this fuzzy layer. I don't know if you can actually see it on this. There's a kind of fuzzy layer around it, and this is the layer what we call the surface mucus layer. And it's kind of like a thick coating. It's polysaccharides mainly on the outside of the coral and in the gut. And it provides a protective barrier, we think, to foreign agents and also changes in the environment. And you can collect this readily. Some corals produce tons of it, some corals not much. And you can collect this surface mucus layer, and we wanted to know, is this like a distinct microbial compartment compared to the direct tissue layers? And then, of course, the skeleton which these stony corals lay down. So for the rest of the talk, I'm going to be showing you data that's color-coded. So when we talk about the mucus, we're going to be talking about teal-colored dots. The tissue will be orange, and then all of the skeletal data will be in purple. And you can see that here are the numbers of samples we're referring to: 207 mucus, 200 skeleton samples. So the first data set that we just submitted for review is done exclusively on these Australia samples. It's 691 samples. It contains 32 genera of corals, 4 outgroups, 2 reefs, and this covers about 17 degrees of latitude. And we wanted to make sure we picked a data set that had a lot of environmental differences. Because if we want to disentangle phylogeny from the ecology of the system, we have to make sure that we have the data set to do that, because it could be that, well, all of these corals are found in one place and it's the environment that's driving those changes. So this allowed us to disentangle these different parts, and it represents about 240 million years of evolution. We did 16S amplicon analysis on the V4 region, and all of this was sequenced on the MySeq samples. And so after some basic stats, each sample had about 14,000 samples, 14,000 reads, and the total data set, we found about 130,000 OTUs. And for those of you not microbiologists, we use the term OTU to represent kind of microbial species. So let's jump right in. So one of the things we just wanted to ask was about diversity. If we look at alpha diversity, the number of taxa that are found across this entire data set, we found a really immediately surprising factor was that within the skeleton, you had much higher diversity of the bacteria that were found in the skeleton across these lineages compared to the tissue, and then actually the mucus was the lowest diversity. We hypothesized exactly the opposite. We thought the mucus was the highest because it's in contact with the environment, that since there are many kinds of environmental conditions, that this would actually have the highest diversity. But in fact, it's actually the skeleton. And we're not really sure why that is, but we find it really fascinating. So for those that like to see PCOA plots, which is the microbial composition pairwise compared across all of these samples, things that are closer together are more similar in their microbial composition. You can see that there are distinct clusters of the skeleton versus the tissue versus the mucus. So these are very distinct communities. That is, not only are they different in their diversity, but the compositionally they're different as well. Some microbiologists like to talk about what is in the core. That is, what microbes are always found in different corals. And this core analysis is based on 70% prevalence. That means for every coral within that category, 70% of them had to have that particular bacteria. Now, real importantly, we only found a single bacterial OTU that was found in every single coral that we looked at. But it's an unknown, which I thought was really interesting. So we just call it OTU1. It is an unknown OTU, and we're trying to figure out what it is. Unfortunately, we could not identify it taxonomically, which is rather frustrating but interesting. And then we also found that there were 6 core members across all of the different compartments. But this is at a very high taxonomic level. It's at the family level, and so you can see there are these names. We often see these in all of our experiments. What that means is that some of these groups might have co-evolved, but we have to be able to test that. One of the things we wanted to see is what drives diversity in these particular habitats. Is it host traits or is it environmental traits? So we had taken a lot of metadata, both from satellite data and also from the data that we collected. And then we did some models to say what particular variables contributed to the most variation explained. And so what I want to point out here is if you look at, in this case, host traits, the tissue was, host traits explain the most of the variability for the tissue. That is, if you look at the host phylogeny, it explained most of the variability of the microbiome in the tissues, where it's less so in the skeleton, but also significant in the skeleton, and less so in the mucus. The largest factor that contributed to the mucus variation was environmental traits, suggesting that the mucus composition of the microbiome is more sensitive to environmental factors than is the tissue. So, for people doing microbiology experiments, if you want to test variables that are interesting in the environment, you might want to look at the mucus and not in the tissue because it seems less variant and it responds less well to the environment, although it does, but to a less degree than the mucus composition. We found a few other things like season were important and which reef you actually analyzed was important. Some of the other things that we found were really important were latitude. So for example, actually was also shocking, latitude was a strong driver of alpha diversity, that is the number of OTUs in the corals, and that was independent of which compartment you looked at. So as you went lower in latitude, you found that there were lower numbers of bacterial communities. At the same time, one of the other variables that we found was very important was colony size. So as colonies get larger, we reduce the number of bacteria that are, the number of types, I should say. So this suggests to us some sort of developmental process. So as corals grow, that's a measure of their age as well. And so as they get bigger and bigger, they're older and older. And so it was probably some sort of, just like in humans, through early development, we have a very variable and widely diverse microbiome in our guts. Older and older, our immune system matures, and it becomes less variable. Beta diversity and alpha diversity are reduced. And so we think the same thing is going on in corals. Well, what about the evolutionary aspect? Are there particular bacteria, as I said, that we always find in different clades of corals? And that can those bugs be contributing to something about the health and longevity of those particular coral taxa? So this is a generalized linear mix model looking at the phylogeny of the bacteria and the phylogeny of the hosts. And so these are all the different factors. So this is host identity by microbial phylogeny, host identity by microbial identity, they're slightly different. Host phylogeny and microbial phylogeny, this is co-phylogeny, this is the one I want you to look at. And then these are all the bacteria and down here. So, and this is geographic area, so this would be environmental factors. So the important thing is that we did find strong evidence of co-phylogeny of some bacteria with their hosts. And surprisingly, the largest number of those are in the purple, which is in the skeleton. And again, this was something we did not hypothesize. The largest evidence of this biggest bubble here, so it has the highest 95% confidence interval, is actually a bacterium that has never really been described in coral skeletons. So that was really exciting, and we're looking that up. And another one here is this guy here. This is a really interesting green algal chloroplast. So when we do 16S, we always get chloroplast. We decided to leave them in there for fun, even though they're not bacteria. We just wanted to see what they were. And this is actually a very unique protist, I should say it's a protist, that seems to have co-evolved with these groups of corals. And then there's a few others like these Endozocomonas. And we call this Endozocomonas-like because a lot of people have evaluated Endozocomonas in corals. It's a common symbiont in corals, and there's a couple genomes out there that have been cultured from corals. But interestingly, the Endozocomonas that we have is distinct from the ones that they've done the genomes of, probably because those were the easily culturable ones, but they're not the ones that are truly co-evolving with corals because they're probably so dependent on their culture. So that's pretty exciting. Now what about resilience? We talked about, do these microbiomes contribute to resilience in corals? And so what we did was we redrew the coral phylogeny here. So this is the complex and the robust clades, both sides. Here we just have the corals and whether they're highly susceptible to disease. We took these disease susceptibility data as a % of disease prevalence based on the literature. Some groups in Australia have put this huge database together on disease, so we took those data and then we color-coded which groups of coral are highly susceptible to disease. And you can see that the higher, the darker the color, the more prevalence of disease. And there are two groups found both in the complex and robust clade, Poecilopora stylophora clade and the Acropora clade over here in orange. And then we superimposed the same phylogeny with our outgroups. So these are the outgroups up here, that's why there's more bars. And then we looked at the alpha diversity of the particular microbiomes that we had from all of these different clades. And you can see some evidence that those that are most disease susceptible also have the darkest color here, which is the lowest alpha diversity, which suggests that if you have very few kinds of microbes present, that you're more susceptible to disease. So it's a very strong correlation. But as you can see, there's some phylogenetic signal here. That is, there's particular groups that are susceptible to disease. So we had to disentangle, again, phylogeny from disease susceptibility. So we ran a bunch of models, and we found that, yes, there is a strong correlation between phylogeny and disease susceptibility, but there is still strong evidence that the microbiome is associated with the susceptibility. Okay, so we did both non-phylogenetic susceptibility measures and a phylogenetic susceptibility. You can see that there's both positive and negative associations with different groups of bacteria with disease susceptibility. So what are those bacteria? Oh wait, well first, I just threw this slide in because someone has asked me about that. What do we know about these groups of corals and why they might be susceptible to disease? Well, some work by many other people, not us, had categorized the different life history traits of corals. Some are considered weedy species, that are very competitive, and one of the things is how fast these individual groups grow. So some corals, like Acropora corals, they go gangbusters really, really fast, and then they die. They die by a lot of disease. So they grow fast, they live fast and die. So as you can see here, there's these slow-growing taxa and these fast-growing taxa, and that microbial alpha diversity and disease prevalence seems to associate with them. So the lower the alpha diversity, the higher the disease prevalence of these fast-growing ones, but the slow-growing ones have higher alpha diversity and seem to be less susceptible. So we think there's a strong relationship between life history, microbiome diversity, and disease. And now we're trying to look into exactly which bacteria are associated with it. One of the bacteria that we always found was in those core memberships of the corals was a bacteria called Rickettsia. Rickettsia are known intracellular obligate pathogens. And we've been doing a lot of work on this because we always find it in our corals. And I wanted to show some evidence here. So we did some fish, and we always find these Rickettsia in these acropora corals. And this is one of the ones we did some, for those modelers here, we did some random forest analysis to see if we could target a single bacterium or two that might be strongly associated with disease prevalence and low diversity, and it was Rickettsia. It was one of them, and Rhodospirillum, which is its sister taxa, so we don't know if they're actually different, they might be in a similar group, were the two that gave the strongest signal. And so we've been finding these bacteria in corals, and they always seem to be associated with disease. That is, if you add these bacteria to among healthy populations of corals, you never find the Rickettsia, but in diseased, they contribute about 50% of the community. So they're heavily associated with disease. And we've been doing some experiments. This bug just keeps popping up. It's like one of those things you just, after you work on something for a really long time, you just keep seeing all these patterns popping up. It just keeps coming up, and I can't ignore the signal anymore. It's flashing in my face. I must work on Rickettsia. So we've done this experiment with acropora in the Caribbean. All of those experiments that I just talked about were in the Pacific. But in the Caribbean, we've done some enrichment experiments and found that under normal conditions, Rickettsia is in very low abundance. But if you add nitrogen, you can increase the abundance all the way to 90%, but on average about 60%. So a huge increase in the number of these bacteria within corals. And like I showed you, they're associated with disease. So we're really to see that this is a really strong connection between looking at the phylogenetic signals of the microbiome and doing experimental data that can kind of link the two. So that kind of brings me to transitioning to our next set of experiments. We've done all this phylogenetic work. We have signals that some bacteria might be important for contributing to resilience or contributing to disease susceptibility. Now, how can we prove that? Experiments, right? I'm really an empiricist. I love to do experiments. I like to go out in the field and change things. Do some construction underwater. That's the best part. So we really wanted to ask, how are changes in large-scale ecological patterns? We know these big changes when you change certain conditions. So if you overfish, algae grows, they compete with corals, corals die. Well, is there some sort of microbiological cascade that occurs at the same time? And maybe one of the reasons these corals are dying is because they're dying from disease. So 8 years ago, when I was an assistant professor at FIU, I met my closest collaborator now, Dr. Darren Berkey-Pyle. He's an ecologist. He works on, he also works in the savanna, but he works mainly on herbivore-algal interactions. And we cooked up this experiment that we call Project Herbivore, trying to link local and global stressors to coral decline by looking at the microbiological level and the microbiological level. So we manipulated overfishing and nutrient pollution, and like I said, in the background there's always climate change. And we hired, again, Dr. Jesse Zaneveld, who helped us from out the biological side. So Jesse is a microbiologist, and he's mainly a computational scientist, and he'd never worked in the field, but I'm a big proponent of, I don't care what your, if you want to understand your system, you better see it, and you better see what's going on. So I taught him how to scuba dive. He went through our scientific diving program, and we put him in the water. And this is a funny picture, and this is day one. He ripped his wetsuit in half on day one. I don't know how it's physically possible to do that, but he did. And so this is a picture of him on our boat on day one. So to do this experiment where we manipulate two important local factors, we started a field experiment out here on Pickles Reef. This is on the southern coast of Florida. So these are the Florida Keys, Key Largo here, and this is Pickles Reef. We did a lot of our metadata analysis from the water chemistry that was nearby. Some important aspects of this reef is that they have very low coral cover. Sad for Caribbean, that is good. 5% coral cover, but large numbers of herbivores or parrotfish because of protection status of those fish for quite some time now. So we wanted to manipulate overfishing, and we wanted to manipulate nutrient enrichment because both of those factors have been implicated in coral decline. So we set up this nested experiment where we built 9-meter squared plots underwater. Here, as you can see, we put rebar down, and then we built these cages. 2 of the cages were completed, and 2 of them were our control, where the top was open. So in this case, fishes can't get in here and eat algae, whereas in here they can still get in. And on the bottom, in this nested part, we added nutrient diffusers, which contain this fertilizer, osmocote, a slow-release fertilizer that will enrich the environment with nutrients, allow algae to grow more quickly, we assumed, and allow bacteria to change. So this is what the experiment looks like from down. So we had these exclosures here, no large fish can get in here, and then we had the open controls here so that there was no issue of building these cages. And then you can't really see them, but here's one, here's a little nutrient enrichment thing. So what happens when you do this? So with the nutrient enrichment, there are little tubes all over, and so you get a perfect distribution of the nutrients all the way across. And we enriched about 4 times the amount of nitrate that's normally there to about 4 micromolar. Right, it says there, about 4 times. So this is what it looks like in an open plot. It's kind of sad if you work in Indonesia. This is a coral reef, I promise. There's a coral, see? And there's another one over here. So this is what it looks like in open plots. If you close them and you keep herbivores out, this is what happens very, very quickly within just 3 to 4 months. You get huge, massive growth of different types of algae. Not only does the amount of algae increase, but the diversity of algae increases. So just wanted to show you that the experiment worked. We ran this experiment for 3 years. And with the individual, we had 80 individual corals. We sampled them monthly for their microbiology. And every quarter, we did all of the benthic surveys. We also did monthly maintenance and care. And doing this for anyone who works in the field every month, rain or shine, was really work. It took about 60 volunteers, and it was like our lives for 3 years. Like Darren and I, this is all we did, I swear, for 3 years of our life was maintain these field sites. So what does the microbiology look like? So this is just one coral, and I'm just showing this to emphasize the amount of data this was. This is a single coral over the course of the entire experiment. And what I want to point you out, each color is a different type of bacteria, and each bar is each sampling set. Okay, so what I want you to notice here is that for the most part, there's a large abundance of this yellow guy, but for the most part, it's pretty variable, but pretty consistent, with a few exceptions. You can see here that in this month, there was this huge outbreak of this red guy. You also see it here. This month, there's an outbreak of this brown guy. These purple and blue guys come up. And so what we noticed is that while there isn't huge shifts, that is, you don't see a massive shift in the coloration on this plot. What you see are a few individual taxa that pop up. And we attribute this to opportunistic bacteria coming in and exploiting the conditions at hand. So it became very problematic to analyze these data, and then we started to think about all of the variables that were going on. So then we characterized the different microbial outbreaks based on different environmental factors that we had calculated. So this is a PCOA plot. This is all of the different individual corals over the experiment. And what I want you to notice is that in the colors that represents the dominant bacterial taxa in that microbiome, and the variable that it was highly correlated with statistically. We're only showing the statistical ones here. So there are some red guys here. These are corals that had outbreaks of Vibrios, or the orange ones, Oscitorles, and these were always associated with temperature spikes in the environment. These guys here, these colors here, were the bacterial taxa that bloomed when corals began to touch algae. That was the variable. We had measured all of those variables. That was the variable they were associated with. And so these were all of the ones that we thought, okay, anytime corals touch algae, these are the guys who might bloom. And anytime it gets really hot, these are the ones you're going to see. And this is everything else. Now, normally, when people analyze microbiological data, they look at people. They supply a stressor. So if this is our healthy population of corals, their microbiome should all look similar. Assume a steady state of a microbiome. And then you might see a shift in that community structure to a different, distinct, stable state of the microbiome. And again, if you have a different one, it may be even further removed from that composition. And this was our expectation. That is not what we found. Oops, I mixed up those slides. This is what our PCOA looked like when we did it the first time. And these are exclosures, so just nutrients alone, just exclosure alone. Nested, and then our controls. I have to say, this was a sad day. This is year 5. of this experiment. We did this and we were like, and we saw the data come out, and I almost cried. There was no pattern. And so I'm like, how can this be that nothing has separated out? But my postdoc is so clever. And he working with data, so he said, there is a pattern. Look, things are spreading out. We didn't see this. What we saw was this. That actually, there's a healthy community, and it has some diversity. And then there were corals that had very widely diverse communities. That is, the variability was very high. And then there were some that were even more clustered, more tightly clustered, the reverse of our expectation. And we started thinking, well, that's probably what's going on. The coral, in this case, we're having a wide diversity. These are these outbreaks. Random things are coming in. The coral can't control its microbiome. It's sick. You have wild variability. And there are other cases where they've died. The community has completely become a single or one or two dominant taxa. And that's what you saw for the temperature stress or the algal stress. They have totally restricted microbiomes. And so we started to model this, and we think, well, so this is our general expectation on a PSCOA plot, that you go from one community to another and to another. And you can measure that by looking at beta diversity. And for those ecologists in the room, microbiologists, we use beta diversity in a slightly different meaning. We're really using beta diversity to look at variability within a community, between sets of samples. So how more variable is one community than another? And so in this case, all these clusters are closely tied together, so their beta diversity is exactly the same. Okay? But you can also have examples like this, where you have one community become highly variable and transition to a new alternative stance to. And that's what this would look like in a beta diversity plot. Or you can have the extreme, where you go from one healthy condition to a mildly stressed, highly variable, to extremely variable community, which suggests that the system is completely falling apart. And so then we looked at our data, and that's exactly what we found. So if you look at the number of macroalgal types that corals were contacting, we found that mortality was associated with this. We also found that disease was associated with the more types of algae. And as corals touch more and more types of algae, their microbiomes become more and more variable. Okay, so they're becoming thicker and thicker, and they can't regulate their microbiome. And you can see that also here. So if you look at temperature, that's the same thing. So just look at the black line. So this is black line. So at very low extreme temperatures, variability increases compared to what normal temperatures are. And then at the highest extreme, you get massive increases in beta diversity or variability in the microbiome, and even in stratus as well. So what we are really seeing here is a totally different pattern that we think is biologically really important, but most microbiologists completely ignore. Most people throw these data out. If there's no pattern in the PCOA, they chuck it, and they say there was no effect of our treatment. So we formed a hypothesis. It was just accepted in Nature Microbiology, called the Anna Karenina hypothesis for microbiomes. So for anyone who's heard Anna Karenina, the very first one is all happy families are alike. Each unhappy family is unhealthy in its own way. That is, when you're healthy, everything looks the same, right? All the corals have the same microbiome, but everybody gets sick in a slightly different way. And that is, everybody's microbiome is different. Therefore, you have high diversity, high beta diversity in the microbiome. And thus, we wanted to know, is this hypothesis true? So we did a meta-analysis of the literature, and it is highly true that for most stressed systems, these are the patterns you see. You see increases in variability of microbiomes just before organisms die. And so, you shouldn't be able to see that part anymore. But anyways, we wrote this paper, and so during normal stress, you have microbiome stability. For example, if you have a viral infection, the microbiome becomes altered, and you get microbiome dysplasia. We're really trying to push this hypothesis. We want people to not throw out their variable data, but to look within their data and see if they see increases in beta diversity, which could suggest that the microbiome stability is falling apart. The host no longer has the ability to regulate its microbiome, and thus new incomers are coming in all the time. Maybe those symbionts are gone, all of this stuff. So we really want you to look at this. Don't throw them out. And in our paper, we recommend certain statistical methods to evaluate this. This should be coming out in a minute. Anyway, the last thing I wanted to point out is in this paper, we found something really, really remarkable. Something we never expected,
but we think it's really, really important. And this really came out because, again, my postdoc is very clever, and he kept seeing these weird patterns. The corals that were exposed just to nutrients, they seem to be different than the corals that were just exposed to overfishing stressors. They both were contacting algae, but those with just the nutrients were dying in a different way. And he noticed that a lot of the ones that were dying just for nutrients had parrotfish bites on them. And so he measured the percent mortality of corals bitten by parrotfish. So under normal conditions, about 7% of all the corals died in this experiment. And those that had parrotfish bites noticed there was no difference, about 0%. But under the nutrient enrichment conditions, 0% of those corals exposed just to nutrients had no predation. That is, they were kept away from the herbivores in the cages. 0 died. But those that were exposed to parrotfish, 62% died. Now, I don't know about you guys, but I never see results like that. So it was so shocking. We checked our numbers over and over again. We had independent people count the bites and confirm the bites visually on the corals. And then we looked at the microbiology, and we noticed something similar. Under just predation, the microbiology didn't seem any different. But when you had the combination of predation via the parrotfish and nutrient enrichment, you got a significant change in composition of the microbes, suggesting that one of the mechanisms behind the mortality was a shift in the microbiome. So to show that visually, basically, under normal conditions, parrotfish like corals. They supplement their diet, but nothing happens to the corals, and the microbiology is nice and happy. If you have overfishing, algae will grow, and corals become sick because they contact algae, their microbiomes die, and their microbiomes change, and then the corals get disease and die. However, under nutrient pollution, you still have parrotfishes. The corals start to grow algae, but of different kinds. The algae contact the corals, and the corals also die, but through a different mechanism. But when you have the combination, this is when you have parrotfish, and the parrotfish bite, the algae grows and the corals die massively. But when you don't have the parrotfish, you still change the microbiology. You have macroalgae, but it's a different mechanism. We think here there's something being mediated by the predation and nutrient enrichment. Either wounding, that is during the wounding event, you have all this nitrogen and phosphorus around, those microbes can grow, they can infiltrate the corals, grow massively, outcompete the coral tissues, and the corals die, right? But here there's no access point. The microbiome is still different, but there's no penetration mechanism, right? So there must be something different here. Is it the parrotfish themselves? Are they vectoring something? Or is it just the wound itself? And that is an experiment we're doing right now on Maria, or Project Tomlin, we're doing with Darren, and to try to understand this mechanism, this change in this trophic interaction between parrotfish, corals, algae, and nutrient forms. So that's where I'm going to end. A lot of people have to think. For the microbiome assessment, my co-PI Monica, and all of my postdocs are here. Everyone has put effort in all of these aspects. My graduate students here, and my collaborator Darren for all of the Florida work and the current Toronto project in Maria. And
you mentioned climate change, so
I was like, oh God.
and it seems like one of the principal threats, of course, to the world is bleaching, especially this second event in Australia. I just wonder if you have any ideas of what the microbiome does with bleaching, and whether there's any compensatory mechanisms to keep those going after they get out.
Yes, we can say two things about that. I didn't show you any of the temperature data other than those two sets of bacteria that seem to be associated with temperature. All of the ecological data that we took seem to associate the highest mortality events during our stressors, so not in the controls, when they were combined with high temperature. So there's a whole part of that paper that is just about thermal stress. So we were lucky during these 3 years of the experiment itself, we never had a major bleaching event. But we did have small
windows of time where the mean monthly maximum temperature did hit above, which is called thermal stress level. And we found that the largest shifts in the microbiome were in our stressor corals combined with those temperature events. So one of the things we really emphasize in our paper, in fact the title of our paper says that global and local stressors interact to cause coral decline, and or cascade down to molecular level and cause coral decline. Because the largest mortality and the largest disease events were in the thermal stress and stressors. But the controls and the thermal stress alone had no effect on mortality and disease.
I guess I'm asking, is there a predictable succession?
Of the microbiome?
Yes.
Absolutely. So last year when the bleaching event was predicted by NOAA, Darren Fab and we were submitting rapid proposals to go out to Maria and to analyze the bleaching event. And I didn't get one, but Darren did. And so we've been working with Darren. He's been collecting samples before the bleaching event, and during the bleaching and after the bleaching event, under normal conditions and nutrient-rich conditions, to see if nutrients increase the prevalence, severity, and longevity of the bleaching event. And producing by talk to see if we're going to talk about that. And there's very strong evidence that it does, in particular reports, mainly in Pastelora. Friday seems to be ineffective. They didn't bleach much. There was a very mild bleaching in Ryan. Bleaching again, bleached last month, and so he extended his experiment. So he's given us all the microbiology samples, and so we've been looking at those microbiology samples. And I would say that I had data for you today, but Pastelora are not the same as everything else we've found. Pastelora are really hard to work with microbiology. The primers can't preferentially amplify that host mitochondria. So we did this whole run, like 600 samples, got one mitochondrial data back. Luckily, they also screwed up their kit, so they let us do it for free to the first thing, but we had to reduce this data. But I know that a lot of people are working on that, and I suspect it's definitely true, that the microbiology is going to shift over the course of the bleaching event. I mean, it just makes sense that the whole entire physiology, the chemical profiles of corals change during the bleaching event. And if their immune status is reduced, you might see a similar thing, that beta diversity increases. We had a student who worked out in Coral-Symbiodinium, and it was devastated by bleaching. There's nothing left. They went back to their field sites, now we have corals to resample. So we have up until the bleaching event, but we have nothing after. So we have no recovery data from the algal corals for that. There's nothing left. I mean, it's like rubble. It's horrible. Christmas Island is devastating. And she sees a similar increase in beta diversity on the way to the bleaching event.
So you showed us the data from the CCS profiles. Have you done measurements and metabolomics as well to look at changes in cheap products that are used through this purification?
Yes, so absolutely. So since my first expertise was in metagenomics, I did a lot of experiments in tanks, changing conditions and looking at the change in the metabolic profiles of the microbes, the potential metabolic profiles. And then what Jesse did, Jesse wrote a program called PyCrust, which is an ancestral reconstruction program. So it takes your phylogeny of your corals, and it looks in the past, and if there's a representative genome from that clade of microbes, it can predict what metabolic potential your microbe will have. And it gives you statistical validation of that based on your phylogenetic composition. So he developed that algorithm, and he used it on our data, and he wanted to see, do the metabolic profiles also shift? And we do, particularly in regards to temperature. Temperature had a huge increase in some categories of bacterial functional groups, like virulence, chemotaxis. Those were the exact same ones that I found back in 2008. So there were very clear patterns that what was done in the lab sort of mirrored the changes that we found in the field. And you can, the program, it's super easy to use, it's not great, but we can help. He's trying to update it. He's a brand new assistant professor, so he's done a lot, honestly. Yeah, you can do this and you see clear patterns. And he also reanalyzed a lot of our old, like TFLP data, and we see similar patterns, like corals that were touching algae, they also have increases in beta diversity. We had never measured that before. We had just done PCOA and we said, well, we see some shifts, but the other ones need more. We be analyzed the data and found something. And he's done that to a lot of people's data and found it's always data diversity is always increasing in these underlyings that are under stress. And so clear changes in the metabolic profiles, the phylogenetic profiles, and the health of the world.
So it's fascinating with biodiversity, and your conceptual diagram had little arrows that were colored to say stochastic. And I wonder, like, do you really think it's stochastic, or is it high dimensional in some sense that could, in principle, be mapped out, but it's just right now too many actors?
Jesse ran some models based on random walk analysis of the data. He did 2 models, I'm going to forget the second one. He did a random walk. So that is if you have a microbiome and you put it in 3-dimensional space and you tell it to randomly walk, where will it go? Right? Will it just bounce back and forth? Will it sometimes move to a new dimension? It actually does on random walk, you can actually get patterns that look like alternative stable states. And then the other one was, starts with a G, and it's a German name, it's very long, it's 2 names. And so this would be deterministic walk, that it's constrained within a certain 3-dimensional space. And so he ran these models to see what would you get if it was stochastic versus directed. And we abandoned that for the paper because since it's a meta-analysis, we didn't want to add any new analysis. So I think he might be working on that. But you clearly see you can get patterns of alternative stable states based on random walks, but it's very rare. The likelihood is very unlikely. But if you have these deterministic ones, you almost always can contain the microbiome within a certain group. You rarely see increases in beta diversity. But you do see it when you have the stochastic. That makes sense. He will probably be able to answer that question much better than
I'm very curious how the communities use your the online tool that you made where people can explore the 16S data all around the world. Could you talk a little bit about that?
Yes, so the only person I know who's used it was Forrest, my own boss. He did, and that was for our phage data. We had done a lot of phage experiments out in Berea where we calculated production rates and things like that, and he asked us for those data, and we just directed it to him, told him which folder it was in, and he used it, and then he acknowledged us. So that's the only example I know of that, but we're always talking about, oh, you can just, and we have tutorials on our website too, like if you want to know how to measure beta diversity, there's scripts that you can just run in on it. So we also have documentaries. We've done a lot of documentaries with us. Documentaries. We have had a few people help us on, I'm going to get it wrong, I'm just going to show my age. It's some social media website where you can get pictures. Instagram. It's not Instagram. So you can look at all of the pictures of every one of our corals. What? It's not Facebook. It's not Pinterest, because that's the one to know. Another one. You can store all of your photos somewhere online, and so when you click on our individual coral, what? Maybe that's it. I think that's it. So you can click on our corals, because some of them we didn't know the taxonomy of. So we kind of put a call, like, hey, look at these pictures, and can tell us what you think without any information. Tell us what you think it is. Huh? It's not on there. I'm pretty sure it's on Picasa. But you can click on it and you can look at it and you can say, how does someone narrow it down? It links to the picture. Okay. So if you click on that sample, it takes you to that and you can write, hey, this is the wrong taxa. Brian was actually just a very good taxonomist, and he got something like 98% right because we confirmed it with when you do 16S, you also get the host 16S. So we use the host 16S as confirmation for our for our visual identification. So that was handy, and he got a few wrong, and a few people corrected him online too. Like, no, I think it's this one. So that was fun. A little social media. Like that. I don't have another account, so probably should have that. I don't know.
Well, if there are no more questions,
You'll join me in thanking Becky again.